meet again in new chapter, Learning Area 5.0, Metabolism and Enzymes. What is metabolism? It is all chemical reactions that occur in a living organism. The example, food is converted into energy in the form of ATP. And second example, formation of carbohydrates, protein, lipid and nucleic acid. We have learned about this in chapter 4. We can divide metabolism into catabolism and anabolism. What is catabolism? It is the breakdown of glucose to produce energy in cellular respiration, and it releases energy. While anabolism is the formation of glucose during photosynthesis, and it absorbs energy. So anabolism is the formation, catabolism is the breakdown. Catabolism break down complex substances into simpler ones, but anabolism is the process of synthesizing complex molecules from simple molecules. Okay, so look at the chart. Catabolism, A, which is substrate, form product which is B and C. Anabolism, A and B combine, become substrate and produce product which is C. This is the process of breaking down. This is synthesize. Okay, now we move to 5.2, enzyme. What is enzyme actually? And why I show you this diagram? Enzyme is an organic catalyst, okay? Mostly made up of protein. It is produced in living cell organisms and speeds up biochemical reactions, speeds up the process in cells. That's why I show you this diagram. Okay, this is an example of enzyme. Enzyme has an active site. It is suitable to one type or specific substrate. After it combines, it will become enzyme substrate complex. This active site is only suitable for one type of substrate only. We look at another example. Okay, this is the enzyme and its active site. So the substrate shape must be suitable to the active site of this enzyme. And different enzyme has different active site. Okay, this is also the example. This is an enzyme and the active site. So this substrate is suitable for the active site of this enzyme. So it will form what? Okay, what is this? We look at the diagram before. Enzyme substrate complex. Okay, enzyme substrate complex. So this is an enzyme substrate complex. The substrate suitable to the active site of this enzyme. Okay, this is only the example. Next, okay, look at the cartoon. Okay, what it said? You are the substrate to my enzyme and nothing could ever denature us. He said, okay, this is enzyme, this is substrate. Denature here means destroy. We use the word denature because in enzyme process we don't use destroy. Usually we use denature. Okay, next example. This is an enzyme. This is substrate 1, substrate 2. Green and blue substrate. Example only. Okay, what he said? It said, sorry. Hey, that is my enzyme. This blue substrate said this enzyme is its enzyme. But this green substrate said, whatever, don't see your name on it. Okay, so do you think this green substrate is suitable to the enzyme? Okay, no, because this shape is suitable to the active site of the enzyme. This one we call inhibitors. Okay, inhibitors. So it will compete with the suitable substrate. And the process in the cell cannot occur if the wrong substrate combine with the enzyme. Okay, I hope you understand about the process. Now we go to the naming of enzymes. How can we know that the word or when you find a word, you know that it is an enzyme, because by adding ase to the name of substrate. So here, this is an enzyme because it ends with ase. It is lactase. And the substrate is lactose. So lactose plus water form glucose and galactose. We have learned also about this in chapter 4. So why we need lactase here? To speed up the process. If there is no lactase, okay, the process can occur, but it's very slow. When lactase present here, the process will become faster. Next, we have many more examples of enzyme: protease, lipase, amylase, maltase, sucrase, lactase, and many more. But why here we have pepsin and rennin? Both also are enzymes, but not end with ase. Okay, this is some enzyme that do not end with ase but end with in. Also enzyme, okay? Next, characteristic of enzymes. Okay, we start from here. Act rapidly. Lead only small amounts in the process. Reusable means we can use it again and again. The reaction is reversible. Unchanged and not destroy. Reaction is specific. Specific here means one substrate only suitable to one enzyme. Have an active site. Speed up biochemical reactions. Need cofactor to work more efficiently. The process can be slowed down or stopped by inhibitors. Next, look at here. We have lock and key. Okay, why? Because we call the mechanism of enzyme reaction as lock and key hypothesis. It means that only the suitable key is suitable for the lock. Because the enzyme, okay, the key here is an enzyme and it has active site or functional shape that only suitable for the substrate to bind with the key. Okay, so we call lock and key hypothesis. Okay, again, key as substrate combined with lock as an enzyme form lock and key complex. Substrate combined with an enzyme at the active site form enzyme substrate complex. Okay, now we have an enzyme here and two keys, good substrate and bad substrate. So which one is suitable for the enzyme? Okay, the good substrate, because the substrate is suitable, its shape here suitable to the active site of this enzyme. This one cannot. This is the complete diagram of lock and key hypothesis. An enzyme and substrate. Okay, this is wrong substrate. This is right substrate. So, specific substrate approach the enzyme, form enzyme substrate complex. Okay, substrate combined with active site of the enzyme. Then, a reaction takes place. Produce two products. Now the substrate separate into two products. The product then leave the active site after the reaction is complete. then the enzyme can be reused. Why we have two arrows here? Because the process is reversible. Okay, reversible. Okay, now we look at the picture. So what is this? Okay, what is this? This is an enzyme. We can do this at home actually. Okay. This is also enzyme projects that I have done in the previous years. Eco Citrus enzyme cleaner and Red Dragon enzyme. So this one is the cleaner we can use to clean the plate, and this one we can drink for healthy body. So both I get from the as enzyme projects. Okay, this is how I produce the Eco Citrus enzyme cleaner. This one what I have done before I get the solution. Okay, we can also use fruit peels or vegetable waste to make natural cleaning products. So all of these vegetable and fruit waste can produce enzyme. So the enzyme can be used as cleaning products, organic cleaning products that cannot pollute the environment. Okay, how to produce the enzyme? Here we call it as garbage enzyme production. It is not garbage, but because we use the fruit peels or vegetable waste, so we use the word garbage here. 1 ratio of sugar or molasses, we use brown sugar. Then put 3 ratio of organic waste into a container and 10 ratio of water. Then ferment for 3 months in airtight plastic bottle. So the process here is fermentation. It will produce an enzyme and can be used as organic or natural cleaning products. You can try this at home, okay? So I think that is all for now. Anything we can discuss in our group later. Thank you for watching.